Hi everybody, I'm Dan. I'm the last speaker of this marathon, so thank you all for hanging in there. I'm here to talk to you about Ascidian Therapeutics. So Ascidian Therapeutics is an RNA editing company. We're developing a treatment for Stargardt disease, and there hasn't been a lot of discussion of Stargardt, at least so far today, for the simple reason that it's been unaddressable by the genetic therapies and the platforms that have come to retinal disease over the last couple of decades. We have a novel form of RNA editing that we call RNA exon editing, which we think can address diseases like Stargardt that have been inaccessible to the field. What we do is harness the spliceosome that lives in each of our cells that's responsible for converting the genetic message from DNA to RNA to protein. And by harnessing the spliceosome with a novel exon editor, we can create full-length functional protein of even in genes that have been too large to package in AAV or have other characteristics that make them unaddressable to genetic medicines. We're the only clinical stage asset in Stargardt disease that addresses the underlying genetic cause of the disease. And because RNA exon editing is not specific to ABCA4, the gene that causes Stargardt, we've expanded the platform in collaboration with Roche to look at a wide variety of neurological diseases. And so I'll talk today about Stargardt, and hopefully as the company grows and the platform deepens, we can talk about a wide variety of diseases both in the retina and in the CNS. So when Ascidian was founded about 4 years ago, we set out to solve 5 key problems in genetic medicines. The first was that most genome editing technologies were focused on DNA. DNA is binary, changes to DNA are permanent. There's not a lot of plasticity, and that creates a safety concern and a need for long-term follow-up that we wanted to escape by editing RNA and not DNA. RNA editing, using other modalities, is very effective at the level of the single base pair, but it's very difficult to edit large stretches of exons. And most folks that are doing genomic editing, either of RNA or of DNA, are introducing foreign enzymes like CRISPR or Cas in their systems. We wanted to avoid that. We wanted to be agnostic to delivery vehicles. We're not a delivery company. We're using precedented delivery provided by others, and we wanted to maintain native gene expression. The platform does this successfully by harnessing the spliceosome to outcompete cis splicing. So I'm going to go a little bit into the molecular biology here. When our pre-messenger RNA is formed, it has both introns and exons. The spliceosome then splices out the introns, puts the exons together, and converts that into the mRNA message. In a disease like Stargardt disease, where you have a mutation in that form, the mutation just comes along for the ride, and the protein that results isn't functional, right? It doesn't, in the case of Stargardt, it doesn't remove the toxic lipofusions from photoreceptors. A RNA exon editor outcompetes the cis-splicing reaction. So instead of a splicing in cis, you get a splicing in trans, where 2 different RNA molecules come together. Now, this is a great theory, and it's been discussed for many years. So let me show you the data that supports that we can actually reduce this to practice and put it into the photoreceptors of our patients. What you're seeing here on the left is a rescue of engineered ABCA4 knockout cells on the western blot. You can see that when we knock in ABCA4, we get nice thick bands. When we introduce a premature stop codon, you see those bands disappear. That's the control all the way on the right. And then in the middle, we restore protein by putting in, these are very early RNA exon editors with actually pretty low efficiency. But ABCA4 is a pretty tricky protein. It's a flipase, transporter protein. And so you can often see ABCA4 on a western, but it doesn't mean it's active. So we worked with the leading academic authorities on ABCA4, Dr. Robert Mulday, and we created the only functional ATPase assay in the field that's actually being used as a release assay for our CMC. And you can see here very nice restoration of active ABCA4. But this is all in a dish. What happens if we go into real human photoreceptors? So we used ex vivo retinal explants These are real human retinas cultured for 28 days with the help of the Lion's Eye Institute in Florida. They come from corneal organ donors. And you can see here that for the first time, we've demonstrated high efficiency RNA editing in a human photoreceptor ex vivo. We're getting efficiency levels between 40 and 50%, depending on the transduction efficiency of the culture. What happens when we take that into an actual living system? So we moved to non-human primates. The RNA scope data on the left is simply demonstrating that we're getting good transduction in the non-human primate photoreceptor. To be candid, this isn't a new finding. Almost all sponsors working in retinal gene therapy get pretty good transduction in NHP photoreceptors. But it's really important that we get it localized to the nucleus because that's where our reaction takes place. And then we can look and say how much editing at the RNA level and how much editing at the protein level results from these experiments. And you can see, again, about 50% editing at the RNA level, about 30% restoration of protein. The bar in Stargardt disease for protein restoration is probably not that high. To state the obvious, it's a recessive disease, so 50% is curative. But in the Stargardt knockout mouse, about 10 to 20% protein restoration provides a phenotypic benefit. In these healthy non-human primates, we're getting over 30% protein restoration. And these are at quite low doses, approximately Luxturna level doses, or even a little bit lower. We're moving quickly into a clinical study. The medical director of that study, Dr. Aliyah Rashid, is here today with me as well, so I encourage you to chat with her about this. We're enrolling patients in a traditional dose escalation design. We'll start with adults. We'll move as quickly as we can into the pediatric population. In Stargardt disease, there is no treatment. There is no asset in the clinic that addresses the underlying genetic cause of the disease. There are only downstream assets. And so we're very excited to lead the field here. And our hope is that using Stargardt as an anchor asset, we can quickly bring RNA exon editing to a wide variety of conditions in the CNS, in the retina, and in other tissues. I want to thank the stellar Ascidian leadership team and broader team that has brought this asset to the clinic. Particularly want to call out Dr. Ehlers, our founder, former head of neuroscience at Pfizer and head of R&D at Biogen. Jay Barth, very experienced chief medical officer, responsible for a couple of INDs and BLAs. Bob Bell also has more than his fair share of INDs under his belt. And somehow they let me come along for the ride as well, for which I'm very grateful. Wishing everyone a great conference and happy to talk more.